Doc Michael.
Good morning, everybody. Um, Welcome to the PAASE webinar for today. It is a privilege for me to be your moderator today. And before we start, I would like to welcome our president, Dr. Marge Peña, to give a welcome remark.
Doc Peña is having internet problems daw po right now.
Oh, okay.
And also, Doc Michael, your sound is coming, ano po, mahina po.
Okay, sorry. Um, is this better?
Yes po. Thank you po.
So maybe I'll just use my... Ayan, March is here.
Okay, sorry, I'm having a terrible connection. My apologies ahead of time. There's a thunderstorm outside. I'm in Missouri, but anyway, good evening, good morning to everybody, and good evening in the Philippines, and good evening to those in the United States. Welcome to our webinar for today. As I have mentioned before, our themes for this year's summit webinars are stemming from the conversations that we had during our Pagtanaw 2050 fireside chats, wherein NAST had defined the directions that we're going and the goals that we are going to achieve in looking back 50 years from now. During that conversation, we were able to identify the goals and what we could change within those goals so that we can incorporate latest technologies and latest advancements in science. And so what quickly became clear to us is that in order for us to get to our vision of Pagtanaw 2050, where we want the Philippines science and technology to be in 2050, we need to identify those individuals who are leading the charge, those who are leaders in the field and can get us from where we are today to achieve our vision. And so that is the theme, inspired by those themes, by those realizations. We have invited and identified speakers who are leading the charge and who are leaders in the fields in all of the operational areas of Pagtanaw 2050. And so our theme then is getting there and operationalizing the goals of 2050. And so for today, we have invited a leader in the field in CRISPR technology that can help us and address a lot of issues related to health and science and biochemistry. And so without further ado, I'm going to introduce to you Dr. Michael Velarde, who is my co-chair of REC 3, to introduce our esteemed speaker today. And hopefully at the end of this webinar, we will have nice conversations and contributions and be able to identify not just those ways that can help us achieve these goals. Because the scientists cannot do it alone. We are experts in our field, but in terms of operationalizing this, we need the help of other stakeholders in our society to achieve these goals. So without further ado, Michael, can you introduce our esteemed speaker for today? Thank you very much and welcome everyone.
Yeah, thank you so much, Dr. Peña. Before I introduce the speaker, I would like to recognize our Vice President of PAASE, Angie Lau is here. Maybe you can just show yourself and say hi.
Good morning, everyone.
Yeah. Excellent, thank you. So I would like to introduce our speaker for today, Dr. Leonard D. And it's very exciting that he will talk about his research on this amazing bacterial defense mechanism that we call CRISPR-Cas. And we know this particular mechanism as the foundation of the modern day gene editing technology right now. Ron is a associate professor at the National Institute of Molecular Biology and Biotechnology, or NIMBB, at the University of the Philippines, Diliman. He leads the Synthetic Microbiology Laboratory and serves as the Deputy Director for Academic Affairs. He completed his PhD under the supervision of Professor Peter Finneran at the University of Otago, New Zealand. Ron's work really centers on CRISPR-Cas biology, bacteriophage-host interaction, and molecular diagnostics. With a focus on understanding how this CRISPR-mediated adaptive immunity, particularly in this Pseudomonas aeruginosa bacteria, and how this defense mechanism evolve, diversify, and respond to the exchange of genetic material between organisms. So without further ado, I would like to welcome Dr. Ron D. to give the presentation. Please help me welcome him as well.
Thank you very much. Okay, just allow me to share my screen and also turn my video. Okay. Okay, it's been a while since I actually spoke to a Zoom audience. So maybe bago lang po. So anyway, so good morning to everyone here in the Philippines, and also maybe good afternoon or maybe good evening to our audience who are in different time zones. So thank you, Sir Mike, for allowing me to share a little bit about our work regarding CRISPR-Cas biology, and also how that sort of this immune system is characterized in Pseudomonas aeruginosa. Okay, so I'm not necessarily well familiar with the audience and how well versed you are with CRISPR-Cas biology, so do allow me to sort of talk a little bit about the CRISPR-Cas first in the introduction, and then I'll sort of revert to our ongoing work in the subsequent slides. Okay, so just to introduce a little bit about our thematics in my lab. So my lab is, as Sir Mike mentioned, is very much interested in understanding phage-host interactions. So we look at bacteria, but we also look at its predators known as bacteriophages. So if you're not known, also bacteriophages are these viruses that specifically infect bacteria. As a virus, they are obligate intracellular parasites that requires a host for them to replicate. What makes bacteriophages rather interesting is that they're always more abundant than bacteria in any ecological space. So they generally outnumber bacteria 10:1, and we estimate that there is this large number of turnover of phage infection, so 10 to the 25 phage infection that occurs per second. So we would expect that bacteria at any environment would be rapidly turned over by these phages. And owing to that has very much stark evolutionary and ecological perspective or importance. So phages are also classified depending on their life cycles. So phages could either be virulent or they could also be known as temperate phages, depending on the life cycle that they possess. So phages could be virulent owing to them having just that one life cycle known as the lytic cycle, shown here at the very outer ring of this life cycle, where in a phage, when they infect a bacteria, they basically rapidly replicate inside the cell and then lyse the cells as a product of that replication. And once the cell lyses, more progenies are released into the extracellular space, and then also those progenies would reinfect more bacteria. There are certain phages that are also known as temperate, and temperate phages can undergo two life cycles, one that is virulent or one that is lytic, and then the other one is lysogenic. So temperate phages are known to undergo this, what we call as lysogenic conversion, wherein the phage could integrate their genome into the host chromosome and then stay there as a dormant prophage. And then as this bacteria replicates, the prophage is also replicated. When the bacteria experience some form of a stress, that prophage can be induced to excise out from the chromosome and then now undergo lytic cycle. So basically, when the cells are no longer permissible for growth, the bacteriophage excises out and set up shop elsewhere in a different bacteria. So the main thematics of my lab that we looked into are these two facets of this interaction. Number one, we basically asked the question, can phages, owing to their lytic properties, can they actually be used to treat drug-resistant pathogens? So hopefully the audience here would be familiarized that there is this huge emergence of drug resistance in our bacterial pathogens, and also this paucity of drug development to combat this drug-resistant pathogens. So phages, owing to their properties that they have the ability to kill drug-resistant pathogens, are now being looked as an alternative for antibiotics. And we ask the questions, can they actually be used can they be developed as treatments for these drug-resistant pathogens? So there are actually numerous studies that looked into these possibilities, and there are some good results that's stemming up from this type of research. But more so than that, what we also need to address is that how does a bacteria actually evolve to bypass this infection? So much like what we know with antibiotic resistance, when we subject a bacteria to antibiotics, it is just natural for them to actually evolve to become resistant. Similarly, if we're going to administer phages as a novel antimicrobial, you should also expect that the bacteria should also be able to evade phage infection and therefore generate some form of immunity. So this alludes to our sort of like the second half of our work, wherein we actually look into this bacterial immune systems. And by understanding this two-pronged or two-angle approach of this evolutionary arms race, this would actually be very instrumental to our designing of phages to be more effective as therapeutics. So one model organisms that we looked into for this type of study is the bacterium Pseudomonas aeruginosa. So we studied Pseudomonas aeruginosa owing to its importance as a clinical bacteria, because it basically infects plenty of these immunocompromised patients and also contributes to hospital-acquired infection. Similarly, Pseudomonas aeruginosa is actually quite notorious for being drug resistant, or its genome is actually quite amenable for the acquisition of plasmids that can confer drug resistance. And there are many instances wherein have extensively drug-resistant Pseudomonas aeruginosa, which therefore confers it to be among the WHO list of priority pathogens. So one part of our study is we're trying to isolate phages against Pseudomonas aeruginosa, but on the other hand, I will be presenting today is how the Pseudomonas aeruginosa resist that infection. So one mechanism that, as the title suggests, one defense system that Pseudomonas aeruginosa possess to defend against phages is the CRISPR-Cas system. In the basic sciences, beyond genome editing, CRISPR-Cas actually forms the adaptive immune system of bacteria. So it is called adaptive owing to the fact that you need a prior infection for CRISPR-Cas immunity to work. So unlike innate immune system wherein you don't need that prior infection for that immune system to be activated. So how immunity of CRISPR-Cas sort of works, it works in these three main steps of adaptation, expression, and interference. But the gist that I want you to get from this slide is that you need prior infection, and then once that prior infection is generated, it would trigger an immunological memory that would generate this an updated memory that would provide this generation of the short RNA that would allow the guiding of an interference complex to its cognate or complementary DNA targets for degradation. And that's how immunity would be imparted in the cell. So our main model organism that I will be describing today is the type 1F CRISPR-Cas system of Pseudomonas aeruginosa. So this is the genome architecture or organization of the CRISPR-Cas system in Pseudomonas aeruginosa PA14, which is the model organism studied across the world. So how this sort of system is organized, so all CRISPR-Cas systems are actually composed of three main components. Number one, the CRISPR array, which basically forms the genetic memory of past invasions. And upstream of this CRISPR arrays, so this CRISPR2 and CRISPR1, upstream of those sequences are the leader sequence, which basically possess the promoter for expression. In addition to the CRISPR array, we have here the third component, which also actually the main component, which is the Cas operon. And the Cas operon basically provides or encodes the enzymatic machinery that imparts immunity. We call this a type 1F owing to the fact that the Cas operon encodes many genes that are expressed as proteins that forms a complex, and then it forms the complex that we know as cascade, which is important for the interference machinery. So just going through quickly how the CRISPR-Cas systems work, just so that we're all on the same page. So CRISPR-Cas basically starts with the CRISPR array. So the CRISPR array possesses this. So CRISPR stands for clustered regulatorily interspaced short palindromic repeats, which means that it is possessed by this highly repetitive sequences interspaced by this very unique sequences known as spacers. These spacers have strong homology to invading genomes or invading sequences known as mobile genetic elements. So the spacers, we would say, are acquired during the course of infections from mobile genetic elements known as plasmids, phages, and also transposons. So the first step to generate immunity is what we call adaptation. And adaptation just basically means that during the course of infection, the adaptation machinery, Cas1 and Cas2, samples the invading genome for very short sequences and integrate that into the CRISPR array, therefore updating the genetic memory of the immune system. So once the CRISPR array is updated, the CRISPR array is transcribed using its leader promoter sequence to this long precursor mRNA that needs to be processed to make the individual guides that will be loaded into the interference complex. So that after biogenesis, certain endonucleases are recruited and binds to the stem loop structure of the CRISPR RNA, so this long pre-CRISPR RNA, to process them to make this mature single guide RNA. The single guide RNA will then be loaded into the interference complex for its surveillance. So during the course of infection, those loaded interference complex would find or match with their targets, so the DNA targets known as protospacers. And then after binding or after recognizing those protospacers, specific nucleases are then recruited on site that would allow the degradation of that invading DNA, therefore conferring immunity. So in the model system PA14 that is heavily studied for CRISPR-Cas biology in Pseudomonas aeruginosa, they basically possess this prototypical type 1F CRISPR-Cas system, wherein in the very middle, you have there your Cas genes or the Cas operon, and then downstream of this Cas operon, we have here the CRISPR-1 and the CRISPR-2. So we would describe it as one Cas operon and two CRISPR arrays. During the start of my lab, what we were actually studying is the strain PA10145. And PA10145 is actually generally used in laboratories for biofilm assays. And it is actually strongly similar to, or strongly homologous to PA14, which is the model strain for type 1F CRISPR-Cas. What you could see there, PA10145 also possess the same type 1F, but the sizes of the CRISPR-1 and CRISPR-2 is actually quite dissimilar or medyo magkaiba, quite different. Unusually, when we were looking into the genome of the PA10145, unusually what we were able to find is that 1.3 million base pairs away from this canonical type 1F CRISPR-Cas operon, we actually also found this isolated CRISPR-3. So we call it isolated because it is far away from its cognate Cas gene. So typically, CRISPR-Cas operons would generally be found adjacent to each other, and that would be the canonical confirmation of how they would look up or how they would appear in genomes. So seeing that there is this unusually, far away CRISPR array, this actually triggered our research. So this work is heavily done by my excellent grad student, so Robbie Paul Malaluwan, who recently defended his thesis just last month, and then he will be graduating in next Saturday. So congratulations, Bobet, who's also in the audience today. Okay, so Bobet's work basically asks two questions. Number one, determine whether or not what is the function of this CRISPR array, so it's this isolated CRISPR array. But more importantly, we were very much interested to understand what is the evolutionary origin of this CRISPR array, knowing that it is in fact functional. So to determine the functionality of this CRISPR array, so we asked the question, can it actually confer CRISPR-Cas-based immunity? So we actually looked at two facets of this immunity, Number one is through interference, and number two is adaptation. So interference means that whether can it actually confer some protection, and number two, after protection, can it actually updates the immunological memory of the CRISPR-Cas system, and how does it do so? So to determine whether or not the isolated CRISPR-3 is in fact involved in CRISPR-Cas immunity, we wanted to actually mimic the course of infection. So at this time, we actually didn't have phages against Pseudomonas. So we actually had to resort to the engineering of plasmids that can be recognized by this CRISPR arrays. So to do this, we actually engineered plasmids that would possess specific protospacers that would be recognized by the individual spacers that are found in the 3 different CRISPR arrays. So just as an example here at the very top, if you could see my cursor, basically we engineered a plasmid that basically contains a protospacer that matches a spacer that is found in the CRISPR array 2. We generated 2 additional plasmids that would have a protospacer that matches a spacer that is found in CRISPR array 1 and CRISPR array 3. So we would expect that if targeting is in fact happening, those spacers can actually recognize those protospacers that are found on those plasmids. So in addition to having just the correct protospacer, we also need, for targeting to be activated, we also need to have the correct protospacer adjacent motif. So this motif is actually used to screen the plasmid first to screen the plasmids, and then before assessing whether or not there is a correct protospacer. So that means for targeting to happen, so the rules of targeting of CRISPR-Cas is you need first a correct PAM, and then secondly, you need to have the correct protospacer or the correct DNA target. So what happens is, what we would expect is if there is a correct PAM and protospacer, targeting happens, and then we could measure the loss of the antibiotic resistance through the loss of that plasmid if in fact targeting is happening. So as expected, what we should also expect as a negative control is that if we actually mutate the protospacer adjacent motif, we eliminate targeting regardless of whether there is a correct protospacer. And then if non-targeting is happening, then we would expect the maintenance of the backbone of the plasmid, therefore conferring antibiotic resistance. And then that is how we would expect that CRISPR-Cas immunity is in fact active in the actual strain. So we first assessed the CRISPR-Cas immunity just by crudely measuring transformation efficiency of those plasmids, whether there is active targeting or non-targeting. So when we transformed Pseudomonas aeruginosa with the 3 different plasmids, we saw different transformation efficiencies. So if there is active targeting imparted by CRISPR array 2 and CRISPR array 1, we saw that transformation efficiency is heavily reduced, so therefore conferring some form of a immunity. If we actually just mutated the PAM of those plasmids, we saw that transformation efficiency is negatively affected. Now seeing that the controls are in fact working and CRISPR-Cas immunity is working, we also tested the plasmid that is recognized or presumed to be recognized by CRISPR-3. And similarly, we saw the same trend wherein plasmids that has the correct PAM are targeted by the CRISPR-Cas immunity, and then just by mutating that PAM, transformation efficiency is negatively affected as well. So therefore, suggesting that the reduced transformation efficiency suggests that CRISPR-3 indeed mediates some form of targeting. So to fully confirm that CRISPR-3 is in fact active or in fact form part of the CRISPR-Cas immunity, we also looked at how the plasmids are maintained across time. And in fact, if targeting is in fact happening, that plasmid would be degraded throughout a longitudinal study. So what we see is that with the CRISPR-1 and CRISPR-2 targeting plasmids, we saw that the plasmids were easily degraded. So if plasmids that were targeted by CRISPR-2, they were lost within 2-day period, unlike CRISPR-1, where it actually took around 5 days for that plasmid to be lost. When we actually subjected the plasmid that is recognized by CRISPR-3, we saw a similar trend with the CRISPR-2 targeting plasmids, wherein upon day 2, the plasmid was totally lost already. When just by mutating that 1 base pair in the PAM region, we saw that the plasmids were maintained across time, therefore suggesting that indeed CRISPR-3 is really formed parts of the CRISPR-Cas immunity of PA10145. Okay, so now that we have established that interference does in fact work in this strain and that CRISPR-3 is also formed part of the CRISPR-Cas machinery, we also want to determine whether CRISPR-3 is also updated through the course of adaptation. So there are two modalities of adaptation, but what we wanted to determine is what we know as primed adaptation. So in primed adaptation, when there is a mutation of the PAM and there is a correct protospacer, what happens is it triggers the phenomenon called as primed adaptation, wherein the Cas adaptation complex, which is Cas1, Cas2, will be loaded into that invading DNA, and then samples near this protospacer form new spacers that will be integrated into the CRISPR array. So therefore updating the CRISPR immunological memory to therefore allowing the targeting of that invader. We can monitor the acquisition of novel spacers across those three different CRISPR arrays by designing primers that would allow the monitoring or the expansion of this CRISPR array. So what we would expect is if CRISPR array is updated just by this course of prime adaptation, by using the same primer, we should expect heavier bands because the spacer is acquired. Okay, so what we were actually quite surprised to see is that in the natural context of primed adaptation, so when there is a mutation of PAM in the literature that should trigger prime adaptation, we actually didn't see any type of primed adaptation in Pseudomonas aeruginosa. However, when there was actual targeting, then that is the only time that we actually saw an expansion of the CRISPR array. So what you could see here, for an example, when we subjected the cells with this engineered vector that has a protospacer recognized by CRISPR array 2. So we should expect that that should lead to targeting. So the plasmid will be lost because of interference, as what we've shown a while ago. But at the same time, as the plasmid is also being lost, the CRISPR array is actually also being updated. And that is denoted by the increase of bands, therefore expansion of the CRISPR array. I won't go through the nitty-gritty of this, but what we were able to show is that regardless of which spacer generated that immunity, all three different CRISPR arrays were actually updated. So what you could see there is that even if they are all recognized, so the plasmids are conferred by, so the immunity conferred by that plasmid is through different CRISPR arrays. However, across those three different CRISPR array setups, all of those setups actually underwent primed adaptation. Okay, so what we were able to show in a nutshell is that yes, the isolated CRISPR-3 forms part of this entire network of CRISPR-Cas machinery, and that we were able to validate this through interference and also through adaptation. So next, the question that we ask is what led to this emergence of CRISPR-3? So plainly, how did this isolated CRISPR-3 came to be? Because it is not necessarily found in the alternative model strain PA14. So to give clues into the origins of CRISPR-3, we first did some comparative genomics, so just assessing where is the closest homologue of, let's say, this CRISPR-3. So owing to the fact that we cannot necessarily assess the actual spacer profiles of the CRISPR-3 because that would contain the immunological memory, we looked into the leader sequence of CRISPR-3. And what we were quite surprised to find is that the leader sequence of CRISPR-3 shares a 98.34% similarity to the promoter of CRISPR-2. So this sort of suggests that maybe CRISPR-2 and CRISPR-3 have some form of the same origin, or likely CRISPR-3 originated from a duplication event of CRISPR-2. However, we cannot necessarily deduce whether or not there's a duplication or CRISPR-3 what actually could be acquired from horizontal gene transfer. So that means CRISPR-3 was acquired through either conjugation or transformation. Okay, so to give more insight as to how well or how prevalent this isolated CRISPR-3 arrays are found across Pseudomonas aeruginosa genomes, we basically mined all of the possible CRISPR arrays or CRISPR-Cas systems across Pseudomonas aeruginosa genomes. So what Bobbitt did is he basically took 1,200 full genome sequence of Pseudomonas aeruginosa from NCBI databases and subjected that for CRISPR-Cas mining. So across this 1,200 Pseudomonas aeruginosa genomes, Bobbitt was able to find this 1,657 CRISPR-Cas systems in Pseudomonas aeruginosa. Predominantly, what you could tell from this this figure is that predominantly Pseudomonas aeruginosa possess the type 1F subtype CRISPR-Cas system. When he further looked into those 1,303 type 1F CRISPR-Cas systems, specifically the CRISPR arrays that they possess, Bobbitt was able to separate them across these three sort of classification. Number one, there are this 800 plus CRISPR arrays that are always found adjacent to a Cas operon. And then there are this 400 CRISPR arrays that are found away from Cas operons. So this away Cas operons could be that they are independently just a Cas operon without a CRISPR array that does not have a Cas operon in their genome, or these are CRISPR arrays that have a Cas operon near a CRISPR array. Okay. So when he probed deeper into this CRISPR away subset, he was able to find this classification wherein this 429 can be subdivided into 281, wherein there is basically a isolated array, and then near in that genome, there is also an intact Cas operon. There are also CRISPR arrays wherein in that genome, there are no known Cas operons. We would expect that this orphan arrays are basically just arrays that are no longer functional for immunity. These isolated arrays are basically what we found in PA10145, wherein there is this CRISPR array, and then 1 million base pair away from this CRISPR array, we have here the Cas operon. What was rather quite astonishing, and what we were actually quite surprised to see, is that all of the 281 isolated arrays really followed what we found in PA10145, wherein its leader sequence in fact matched those of the CRISPR array 2, therefore suggesting that there is that common origin between CRISPR array 2 and CRISPR array 1. Furthermore, when we actually looked at the localization, so the specific genomic loci of where these CRISPR arrays are found, we actually found that all of those isolated arrays are always found at the same position in the genome, regardless of which Pseudomonas species they were. So this led to us to the following findings, that the isolated arrays are actually frequently occur in Pseudomonas aeruginosa, and most of these arrays reside specifically at this precise loci, this YBAK MMSR region. So this led us to actually make a lot of more questions, such as why is it always there, and why is it always precisely at that position? All of this isolated array seems to have also these upstream sequences that are highly similar to this CRISPR array 2, therefore denoting that common ancestry or common origin. However, we cannot necessarily deduce specifically whether or not that the CRISPR-3 array came from a duplication or could have been acquired laterally through horizontal gene transfer. So this led us to a question, could CRISPR-3 have emerged from a recombination event involving CRISPR-2? Whether it is through duplication or through horizontal gene transfer. So one of the clues that actually led us to our specific model is the specific enrichment of mobile genetic elements that cluster next to the CRISPR array 3. So when we fine-tune specifically this specific localization across the Pseudomonas aeruginosa genomes, what we were actually able to find is that there is this enrichment of mobile genetic elements that cluster next to the isolated CRISPR-3. So you can see here, there are this orange boxes are the isolated CRISPR-3 at this precise locus, the YBAK MMSR loci. And then what we would see here are specific instances where there are prophages that are associated also within this precise loci. In addition to prophages, what we also found are these transposable elements, and shown here in pink and blue, that are also found specifically at this precise loci. So this sort of led us to this hypothesis that this mobile genetic elements likely confers the ability to basically transport this isolated array at precisely at this genomic loci, and therefore allowing the ability of this Pseudomonas to be able to transport this isolated CRISPR-3 across genomes. Okay, so this highly transmissible MGE, such as prophage and integrative conjugative elements that we were able to find within CRISPR-2 and also CRISPR-3, likely facilitates its transmission across genomes. So to further generate proof that the isolated CRISPR-3 is literally horizontally acquired, we therefore looked at the spacer profiles and also compared that across the genomes. So the spacer profiles of the CRISPR array sort of could act as a barcode and therefore denote, but the similarity of the barcodes should confer some form of a common ancestry, because those barcodes or the spacers found on those CRISPR array would be indicative of their past immunological, of the past history of the invasions. So when we compare, just by looking at when we compare the spacer profiles of CRISPR-3, what you could see there are, CRISPR arrays that are highly similar to each other across different genomes, and therefore would denote some form of a common ancestry. We also did the same thing. So again, what we wanted to do is to try to decouple the evolutionary ancestry of the CRISPR-3 and those of the CRISPR-1 and CRISPR-2. So when we created a phylogenetic tree here in the left for CRISPR-3, we wanted to compare that with the phylogenetic tree of CRISPR-1 and CRISPR-2. So if the hypothesis is that CRISPR-3 is horizontally acquired, therefore, the memory or the profile of the CRISPR-3 should be sporadic, regardless of which CRISPR-1 and CRISPR-2 that the genome actually possesses. So when we combine these two phylogenetic trees to actually look at whether horizontal gene transfer is in fact happening, we first constructed the phylogenetic tree, the inner ring for CRISPR-1 and CRISPR-2, and then we embedded the phylogenetic tree of CRISPR-3 on top of the phylogenetic tree of CRISPR-1 and CRISPR-2 to determine the evolutionary linkages between the two. So just by narrowing it down to just one subset first, so here highlighted at the 6 o'clock of the tree, what you could see there are these instances wherein the CRISPR-1, CRISPR-2, and the CRISPR-3 are all following what we would expect from sort of like this vertical transmission of ancestry, wherein the CRISPR-1 and CRISPR-2 are regularly updated. Similarly, we see the same type of ancestry with CRISPR-3. So in this subset, what we could expect is that those strains follow the vertical mode of transmission. However, if we're going to look at the actual phylogenetic tree as a whole, what you would expect is that the CRISPR tree is actually quite sporadic and sporadically distributed across the phylogenetic tree. So there are this blue here, but you could actually see the blue on the other side of the phylogenetic tree, which therefore suggests that the isolated CRISPR tree is more horizontally acquired rather than vertically acquired. So just to prove this further, we actually saw certain instances wherein, number one, we actually saw the CRISPR-3 that are very much the same, but they actually possess two different CRISPR-1 and CRISPR-2. And then similarly, we found the same CRISPR-1 and CRISPR-2, but they possess two different CRISPR-3. So all of this phylogenetic analysis, just by looking at the spacer ancestry, suggests that the CRISPR-3 array independently evolved from this adjacent CRISPR arrays, and likely owing to the association of mobile genetic elements with CRISPR-3 and also CRISPR-2, likely result from this horizontal dissemination across the Pseudomonas aeruginosa genomes. So actually, just to conclude everything that I've talked about, so I guess what's the importance of our data, I guess? So again, looking into the picture, the broader context of the possibility of the use of phages against Pseudomonas aeruginosa, we first need to understand how a host defends against those viruses. One of the mechanisms is through CRISPR-Cas-mediated protection. However, unlike the conventional thought that CRISPR-Cas, for you to generate an immunity, you need prior infection for you to be immune against that phage, what we were able to show is that in the Pseudomonas aeruginosa genomes, there is this instances wherein CRISPR array can actually be shared across populations. So through this transmission or through horizontal gene transfer, isolated CRISPR array can be transmitted to another recipient and therefore providing an immunity to the recipient cells even without the context of infection. And therefore, that cell would now be immune to that phage, even without infection. So the implications of this and why this is the case is that in some populations, what they want is having an intact CRISPR-Cas would actually prevent horizontal gene transfer. If you prevent horizontal gene transfer to occur, evolution would slow down. So in this instance, wherein populations can actually share CRISPR-Cas immunological memory, then there will be certain instances wherein cells would no longer have an intact CRISPR-Cas systems and therefore restore the influx of horizontal gene transfer. Altogether, what we would expect is that the population are actually sharing this immunity and therefore conferring some form of a pangenomic immunity. Yeah. So altogether, what I would hopefully I was able to impart that there is some form of rewiring that the population of Pseudomonas can actually confer just by actually transferring CRISPR-Cas or CRISPR arrays throughout that population and therefore mediating some form of a populational defenses. So I'll be happy to answer questions if the audience have questions, and hopefully that give you an insights into our ongoing work in SML. Thank you very much for listening.
Thank you so much, Ron, for that wonderful talk. We can now entertain questions. You may leave your questions in the chat box. You can also raise your hands and unmute yourself if you would like to ask question directly. Let's see if there are any questions so far. So while we're maybe, if not, maybe we'll. Yeah, go ahead.
May I ask a question?
Yes, please.
This is Asuncion Raimundo. Hi, Ron.
Hi,
Nice to-
ma'am. Hi, ma'am. Morning po.
Good morning. So the mechanism that you described, is that applicable to other pathogens and to other microorganisms? I was thinking of the application, I'm
Yes.
involved in bioremediation. I thought since CAS CRISPR, CAS-mediated mutants are not considered genetically engineered, right? So they are not banned from being used, being applied. So can that be used for a bioremediation bacterium that will widen the scope of its effectivity in the environment?
Okay. So in the context of bioremediation, ma'am, so if I understand your question correctly, so can we actually make more phage-resistant bioremediation bacteria? Is that correct, ma'am?
No, actually, wider scope. It can attack more heavy metals, for example, just mercury can attack also lead, or other heavy metals. What do you think?
So can we engineer a bacteria that will be more important for bioremediation? I mean,
sort of, right.
So I mean, just to give a bit more context of our work now. So so the specific context of our work, so the isolated arrays, ma'am, yung sharing of immunity, when we actually expanded our search whether or not this is applicable in other bacteria or do other bacteria participate in this sort of sharing of isolated arrays, we actually didn't see the similar, parang we didn't see that same phenomenon. So what we are able to show is that parang this is mostly the model system. The model that we're providing is very much only found in Pseudomonas aeruginosa. However, there are some instances naman in other bacteria and other model organisms naman wherein they've seen that the entire CRISPR-Cas systems can actually be transplanted through horizontal gene transfer. So this is a sort of a recurring thematic, but there are some nuances that makes it specific towards individualized species of bacteria po. So parang similar thematic, different yung specificity.
Okay. Think about it.
Yeah.
Thank you.
Yeah. So I mean, I couldn't really try to address how it works to make it parang the engineering perspective for other parang biotechnologically important bacteria.
Right.
But in some way, that would still require some form of engineering po kasi. So some form of a GMO.
Yeah. Let's talk about it some other
Yes
time.
po, yeah. So in PSM po, ma'am, I'll be there. We can talk about it.
Yeah. Okay. Thanks, Michael.
There's another question by Dr. Bontok. Maybe you could check the chat box. They're
possible to
asked.
create, yeah, is it possible to create, so the question by Dr. Is uh, is it possible to create super mobile elements for a bacteriophage in order to annihilate Pseudomonas aeruginosa? Yes, especially those which are drug resistant can give approach to other bacteria. Yes, so the answer there is yes. So the new aspect of how we should think about phage therapy or the utilization of phage to combat drug resistant pathogens is we actually need to take account the immune system that are already found in those bacteria, and we should basically engineer. So basically, if we're going to look at therapeutics, we already need to think about what should a phage possess to disarm the host, their immune system. So there are current studies that showed that they actually engineered phages to possess anti-CRISPR proteins. Those anti-CRISPRs would basically neutralize the native CRISPR-Cas of the Pseudomonas. And then by equipping the phage with those anti-CRISPR proteins or this anti-CRISPR gene, basically they provide a more long, like a more effective uh, uh, antimicrobial, I guess, lytic response. So they would kill the host cell much better, and then immunity would actually not, so immunity against the phage will actually not be, they were not established because they were very more potent. Thank you.
Thank you. Any other
I
questions?
have a question. Can I ask
Yes,
a question?
please.
My name is Ben Texon. Uh, Dr. D, I'm impressed by your work. Since it has therapeutic possibilities, have you thought about the implications regarding uh, having patents on it? Because remember, there were the very famous work done at Utah, and
Oh, yeah,
we
that's
two
correct.
Filipino scientists found a painkiller, and the patent was made by another person in their laboratory and not these two Filipino scientists. So my question is, if you considered it and talk with people knowledgeable in the patents.
Ah. So, we haven't really reached that point yet. I don't think we have things to be able to patent yet. But so we have two things now in our lab. So one is the collection of phages against Pseudomonas aeruginosa, and then we also now have this preliminary, at least this study we're in, specific strains of Pseudomonas aeruginosa, and then we know how the immune system sort of work against them. uh if we're going to basically go for that patent, we basically should engineer the phage that we have and then make them really effective in killing the barrage of Pseudomonas aeruginosa. If we're able to find those and also establish those, then those should be patentable. At least the combination of phages should be patentable. But yeah, we're not there yet, but hopefully we'll get there, Bob.
Okay, I'd like to make another comment.
Yeah, Bob.
One of the people was in the same lab with me, his name is Billy Freitag. He had ano lang, drug candidates. He had a company called Mayagen. Drug candidates pa lang, which he sold to Gilead for
Hmm.
$2.5 billion. Drug candidates pa lang.
Sana
Okay.
po sa
So,
pabilis, pero sige po.
but then when he sold this company to Gilead, he remained working on it.
Oh, okay.
And yet his company got $2.5 billion. So anyway, look at that angle. They
Yeah.
don't necessarily have to have a product. All you need is a drug candidate. Because I don't know if you're aware of it. In the world now, we only have about 18 to 24 new drugs every year. So
Correct.
yours is potentially a very exciting possibility. So anyway, I wanted to tell you that it doesn't have to be complete. It
Okay.
can only be a drug candidate. Okay?
Okay
Thank
po.
you.
Okay. Thank you po. Thank you for
Yeah,
that.
thank you so much. And I see academician Giselle here, Concepcion. She has a
Hi, Ma'am
question
Gisela.
also.
Okay, thank you. Thank you, Michael, and congratulations, Rondi, particularly for your work on the evolutionary aspects of the new CRISPR that you discovered. And that's got many implications in nature because in our work, we have found Pseudomonas that is free living,
Yeah.
and then we also have Pseudomonas that is symbiotic and producing a lot of bioactive compounds. And then, of course, we have the Pseudomonas that is a nosocomial, you know. infection. Okay, so in relation to that, I just like to clarify what Ben mentioned. He's referring to the discovery of Baldomero Oliveira. But the patent that was filed for that has made it big, is on its anti-pain effect. What was published by Baldomero Toto Oliveira and his group was first on the conopeptide action on calcium channels, blocking calcium channels in relation to stroke. So the application to pain was not discovered at the time in Toto's lab. So anyway, it's a long stretch. We're talking about small molecule drugs, peptide drugs, but in this case, I think Ron is talking about a different type of therapy. Okay.
Correct.
So, anyways, going back to your particular topic. So, well, I tried to recollect or remember some of the knowledge that I know, but of course, I also try to AI all of these things just so I'm not talking, uh,
Yeah.
out of the box or whatever, So, the strain of Pseudomonas that you used is, I think you said biofilm forming.
Correct. The
Yes,
PA10145. Yes, po.
biofilm forming. Okay. So then we have antibiotics that can disrupt biofilms. And then a lot of these drug resistant pseudomonas or staphylococcus, they're actually because of the biofilm.
Correct. Yeah.
Okay, and it's nondescript. You say it's drug resistant, but it's because the small molecule drug cannot enter the cell because it is covered with the biofilm. The bacterium is trying to behave like a multicellular animal, right?
Correct. That's
Okay.
right.
Anyway, so this is what I wanted to know about your first part of the talk on
Mhm.
interference and adaptation in relation to your designed. Is it a
Mhm.
plasmid? It's not a bacteriophage. What did you design? And what is the antibiotic that you use? Is it a general antibiotic that is known to disrupt biofilms, or is
Mhm.
it a cytotoxic one? What is its a brand of antibiotic, and how does it
Mhm.
act? Okay,
Yeah.
because I think that connection has to be established
Mhm.
also in your talk, if I may. I know this is a very short talk, and I'd like to share here the comment or suggestion of academician Sean Raimundo, Doc Marge, and VP Angie, President Marge. When we host our PASA talks, let us partner with the professional societies. So in this case, sana, we had the whole MCP or MSP with us. Or if it's a chemistry talk, maybe, chemistry. Or if it's a molecular biology and biochem, PSBMB, so that we have a wider audience. I think it's about time for us to widen our reach. So thanks, Ben, for joining us because you are a PASA Vanguard member, right?
Mhm.
Okay. So, anyways, those are my questions. How did you design your plasmid? Okay, that would trigger the interference and confer antibiotic resistance. The
Yeah.
storyline that I would like to understand better.
Okay,
I mean,
ma'am.
it may be basic. You may have presumed it. I'm sure it is in the thesis of Robbie. Is this a PhD thesis or a master's thesis?
This is a master's thesis. So actually, ma'am,
Fantastic.
yeah.
Yes.
So there were plenty of things that I couldn't really present.
Yes, Ron. But I really look forward to hearing you. Thank you so much.
Okay. Just to address that, ma'am. So actually, ma'am, I
Okay.
have a different student who's studying biofilm formation of that strain. So actually, it's a different total mammoth, no? So we actually because we wanted to determine parang what we want to determine how this biofilm sort of work in different facets of. So I mean, although it is established in Pseudomonas, but we wanted to probe deeper. It's like can we actually find different angles of regulation that triggers biofilm formation? But that's a different mammoth. For this mammoth, we wanted to study is based. So we didn't really have phages at the time when Bobet started to be able to mimic the course of infection. So again, so just if I go through here, So the idea is, so if immunity is actually happening, then what we would expect is a phage. If a phage infects and then it has a protospace, or it has a target that is recognized by one of the CRISPR array, then immunity should be imparted. But because we don't have that phage, we basically use plasmids instead. So those plasmids should be like a common plasmids that we could easily introduce to Pseudomonas aeruginosa. So the one that we use is basically contains a streptomycin resistance. So that means the plasmid should confer, if the plasmid is maintained, the plasmid should confer streptomycin resistance to Pseudomonas aeruginosa. What we then did, so if a plasmid would be recognized by this CRISPR-Cas system, we basically just introduced like a short sequence that would allow it to be recognized by any of the CRISPR arrays. So for example, here, we show here the CRISPR array 2 here. So if you see my cursor, in the CRISPR array 2, the second spacer there would basically possess the sequence. What we basically just did is we introduced the same sequence to this plasmid. And then what we made sure is that it must have this correct PAM. If there is that correct PAM, then targeting will happen. Then if targeting happens, that plasmid should be lost. And then if the plasmid possess streptomycin resistance, then the Pseudomonas aeruginosa should be streptomycin sensitive. So ganun lang po siya. We couldn't really address the biofilm formation because everything that we did is basically in liquid culture. So we didn't really do a lot of the biofilm. So we couldn't really put that into context whether biofilm is in fact imparted. But we do know that the same regulational axis that controls biofilm also controls CRISPR-Cas immunity in Pseudomonas aeruginosa.
Okay,
Can
thank
I
you. So
take one more
that's
comment?
yeah, that's just I'll just respond to that, Ben. No?
Yeah.
So that's what I think I would like to point out that these experiments on of course the genetic level would really be more meaningful, or would be well, more complete, uh, go towards completion if you yes, do the right functional experiments, you might say, on the physiological and the you might say biochemical level, or microbiological level. So in this case, now it's clear it was not the biofilm that you were testing. It was a liquid culture. So it was a broth culture.
Correct.
Broth. It's just a quantitative. So
Yes,
you're able to quantitate,
that's right.
because you don't anymore do agar based culture. We do right, broth culture, so that you can measure antibiotic activity or resistance quantitatively, right? Did you do those responses of your
Yes,
antibiotic? Wonderful. So anyway, those things make your uh, work even more, a lot more meaningful. So in the end, you're going to wonder what it is that was produced, was produced or not produced by the microorganism. And as I said, the Pseudomonas is a very rich pangenome. We know it. And a lot of that pangenome is also dedicated to secondary metabolism.
right.
Okay? So, anyways, thank you so much. Thank you
Thank you.
so much, Ron.
Can I make a comment?
Go ahead, Dr. Ben.
Yeah. The comment I wanted to make has to do with documentation. Two things to consider. The moment you publish this information in a public journal, you've lost control. That's something you have to keep in mind. The other thing to keep in mind is you have to have notes, the documentation in a logbook, what you have done, better with explanations of why you did it, how it was done, the results, and so on. Because when you go to the patent process, the documentation matters a lot, and the competition is incredibly horrible. Yung mga lawyers, particularly yung mga developed countries, they will try to swallow you. So importante that you document it very, very well. You have to be careful. Even at this early stage, may I suggest you see a good patent lawyer. So you protect your intellectual property. Like I told you, that guy who was in my lab, we shared the same lab. He made a ton of money out of it, but he was careful. But like I said, he sold it to a larger company, but he continued working on it. You can continue, have your cake and eat it too. Okay,
Ano po
sige,
ba
that's
nakabili?
my
Sige po.
two cents worth.
Sige po.
Thank you. Thank you so much for that advice. There's another question from Stephen Paul Ortia.
Possible to touch. Yes. Ideally, technically, yes, we could apply the same approach. So for example, if you're going to use phages against carbapenem-resistant organisms, or let's say Pseudomonas, so as long as the phage can infect, if you know the molecular mechanism of infection, then generally you could infect the same phenotypic or drug-resistant strains. That's not okay lang.
Okay. And then the next question, may follow-up question then si Dr. Bondok.
You mentioned that bacteriophages outnumber 10 is to 1. So theoretically, there are more. So that's correct. There are more. Where are they hiding? They're basically there. They're basically in that same space. So are they undergoing continuous evolutionary mutations? Yes, that's actually correct. So we call this evolutionary arms race, and that the time scale is actually very quick. Infection happens, but because of the negative selection, bacteria immediately generates mutation that would bypass infection straight away. The easiest mutations that bacteria would do are mutations on the host receptor, therefore eliminating um, recognition of the phage. And then, beyond, it's a continuous cycle, and therefore we call this the evolutionary arms race of the phage and also the bacteria.
Any other questions from the audience? Maybe you can entertain one, two questions. Maybe I'll ask a quick question lang regarding the CRISPR. So there's now CRISPR-3 that you found. So why do you think that the bacteria needs it? And is it because that the bacteriophage maybe is able to adapt also?
Yeah.
I mean,
So right. So I think we must look at it in the perspective of the bacteria, not necessarily of the phage, because it's the bacteria that undergoes this evolution. So let's say if I'm a bacteria, let's say we're all here, we're all sharing that same immunity. But at the same time, what we must recognize is that bacteria would need to undergo some form of horizontal gene transfer for them to evolve much rapidly. Diba? So if I have a very robust CRISPR-Cas immunity, I actually would be less evolutionary favorable because I would be acting against mobile genetic elements. So in this scenario, I think what we see now is that since not all bacteria possess CRISPR-Cas, then that means there's some negative effects to possession of a CRISPR-Cas. So if we're all that same population, then we need to have that mechanism to sort of maybe balance horizontal gene transfer and also having possession of an immunity. So to do this, a bacteria could basically share CRISPR-Cas, so basically transmit CRISPR-Cas entire population, or in hindsight, actually just share your immunity. So in that, there would be scenarios wherein I would just have an array. There would be a scenario where I would only possess the intact CRISPR-Cas, and there will be a scenario where I don't have an intact CRISPR-Cas. So those that do not have the intact CRISPR-Cas can undergo evolution, and then when it is actually more favorable to have CRISPR-Cas, I could just basically transmit my entire CRISPR-Cas operand to the population, therefore protecting some, if not all. Parang ganun yung nangyayari. So parang what we see is that sort of this bet hedging scenario for an immunity. So this is what we sort of this is our model for what's happening. And it's basically this tempering of horizontal gene transfer and immunity. And one of the mechanism for this is just by sharing this CRISPR array.
so it looks like Dr. Giselle also has another.
Quick one.
Ah,
So this is Ron, this is an ATCC strain of
correct.
the Pseudomonas. So
Yes, ma'am.
to a certain extent, it's a stable, stabilized strain.
Correct,
So I'm
ma'am.
just curious whether this CRISPR-3 is something that would be found in nature and not
Ayan.
just in ATCC strains.
Ah,
And have you checked it over passages?
correct.
Okay, thank you.
So we actually so we were very curious kasi parang it's not found in PA14, which is the actual strain studied for yung mga clinical strains. So that led to the comparative genomic studies, but also we actually sourced a Pseudomonas strain from PNMCC. And then when you sequence the genome, it does actually in fact contain the isolated CRISPR-3. So it is found elsewhere. So yun naman po.
There's a question from Dr. Vic Ilag. Looks like he's asking about the mechanism on horizontal gene transfer.
We have an idea what mechanism. So there are three paths for horizontal gene transfer: so transformation, conjugation, and transduction. So we would expect, I mean, expectations lang. I mean, if I would just give a educated guess, it would likely be in Pseudomonas would likely be conjugation or transduction. So in this slide, what we were able to show, so I mean, just to give some more inference, at least context for that answer. So we were able to find so not only prophages, but we also found integrative conjugative elements that are next to the CRISPR arrays. So owing to that, it seems likely that it is through those conjugations, so the conjugation of integrative conjugative elements, and also the movement of those prophages out by excise out of the genome and into another host. And then it's through that erroneous or maybe does that parang coincidentally napapackage nila yung isolated array tree, and that is how it is transmitted from one genome to another.
Thank you so much. Dr. Gisela, do you still have any further comments?
No more. Thank you and congratulations. Maybe one last is UP takes care of the filing of patents. So Ron is aware of the process, and it should be improved much more in this new under this administration, Ron. And let me just say that in the Philippines, the policy is still first to discover. Okay, first to discover. So in that case, the notes that Dr. Ben refers to, they're very important. But in the United States and elsewhere in the world, it's a real race. It's already first to file. Okay, first to file. So it's really a very competitive. And in the US, we know there is a 1 year provisional patent, after which you should work very hard to be able to file a full patent or not. Okay, but here in the Philippines, it's a very slow process, and patent review is, I must say, I have gone through it. So many compounds, Ben. I would not say that the review is competent. I'm telling you. So it's really tough for us, Ron. But on the other hand, you do not yet have a specific entity, and I do not know that the first part of your talk, which made use of, as you say now, a plasmid, some common plasmids, and then it's streptomycin. It does not really uniquely identify a potential therapeutic strategy or product, because as you said, it's until now bacteriophage therapy is still just a possibility. Kasi mahirap no? We know that many are still looking into small molecule drugs, but I'm not discounting this possibility at all. So once again, congratulations, Ron, and more power to you. Thank you.
Thank you so much. So looks like we're at the end of our webinar, and I will turn over to Angie. Maybe you have something to impart.
So I'll do the closing remarks. So on behalf of PAASE, thank you all for attending and joining today's event. And of course, thank you, Ron. This is really a very insightful and engaging talk. So as you can see from today's lecture, Dr. D showed us the understanding of how bacteria acquire and rewire their CRISPR defenses is some key to redesigning pages that can become some effective therapeutics in the future. And during the discussion, it's really a lively discussion. And we see how many of the researchers or the audience here are really excited, and everybody wants to help Ron push his research forward. So it was really wonderful to see how Dr. Steve's work spark ideas and conversation across disciplines. And this reminds us that answering these fundamental biological questions can open new possibilities for addressing the growing challenge of especially for antimicrobial resistance. So thank you, Ron. And of course, I'd also like to thank our REC chair, Dr. Michael Velarde, for guiding us today's discussion and for organizing this talk. So before we end, I think Michael will award a certificate later, and then we'll also like to promote and invite everybody for our next session on July 10 that will feature Dr. Ong Lau, Dr. Mario Ong Lau. He will talk about the key technologies and scientific roadmap of Kronos toward sub-HA gravitational wave detection. So we also expect and hope to see you on our next webinar. So I'll give the floor back to Michael.
Oh, the certificate of appreciation is given to Dr. Rondi. It's flashing this screen. And this is on behalf of PAASE. We're thankful that you're able to share your expertise on this research of yours in CRISPR related to CRISPR presented this day. This is on behalf of our president, Dr. Marge Peña. Looks like Marge is still here.
Yeah, I finally got my connection back.
Oh, excellent.
We should have a photo op as well.
Yeah, let's do that. Could you please turn on your cameras? And maybe we can remove the share screen so we can see everybody. Okay.
Angie, are you doing it?
Okay po. 1, 2, 3. Another one po. 1, 2, 3. Page 2 po. 1, 2, 3. Smile. Okay na po.
Okay. Thank you so
Thanks,
much,
Deb.
everybody.
Thank you, everyone. Thank you, Michael.
Oh, thank you. Thank you.
Thank you, Michael. Bye-bye.
Bye.